Good morning, student. Today our topic is restriction and modification system, and we will also discuss about what is called a linker and adapter. We'll start with little bit information about the discovery of these restriction enzymes. The first restriction enzyme was isolated in 1970 by Hamilton Smith. The name of the enzyme was Hindi 2. He was done the subsequent discovery and characterization of numerous restriction endonucleases. From then, over 3,000 restriction enzymes have been studied in details, and more than 600 of these are available commercially and are routinely used in DNA modification and manipulation in laboratories. Now, before 1970, there was no method for cleaving DNA at a discrete point. All the available methods for fragmenting DNA were non-specific, like mechanical shearing, intense sonication with ultrasound. That also can break the DNA, approximately 300 nucleotides, and can be and also mechanical shear can also be achieved by high speed steering in a blender. These type of techniques will cause the breakage of our DNA in a random fashion, which was not that much used in gene manipulation technique. So the discovery of these restriction endonucleases and the DNA structure these two are the milestone in recombinant DNA technology. Now the first enzyme, so called before 1970, the first enzyme which was discovered 1968 by Methylation and Yan was from E. coli K12 strength. Okay, but unfortunately that enzyme specificity was not so correct, and it was recognizing some sequence and cutting in 1,000 base pair apart, so that didn't become so much popular. The main breakthrough came in 1970 by Hamilton Smith discovery, the enzyme name as Hindi 2. Now, restriction endonucleases, it cleaves a specific DNA sequence, protect bacteria, actually the first isolated from bacteria, so extensively studied in bacterial system. So it protects bacteria from phage infection by digesting phage DNA after infection. Cellular DNA, but at the same time, E. coli's own DNA is protected by some other mechanism. Otherwise, the question is that if a bacteria is having a restriction endonuclease among themselves, then why not it is chewing its own DNA? The answer is that this bacteria have some other protection method. They methylase, DNA methylases are there. They methylate at a specific position of their own DNA, and that's how they block the restriction enzyme activity. The small cartoon is showing the yellow color bacteriophage is actually injecting its DNA, which should be treated as a foreign molecule and will be recognized by the bacterial. This is the bacterial host whose own DNA is red color and which is all MM written, meaning methylated, and will be recognized. The red color DNA which is coming from bacteriophage will be recognized as a foreign, and with the help of the restriction endonuclease, that will be digested, and bacteria will be safe. Now, if this scenario is the correct one, then bacteriophage will not exist in our world. But that is not true. Bacteriophages are there. Now, what is actually happening? Here I'm going to show you that this restriction and modification system is a host controlled restriction and modification system. Let us describe by using two different E. coli strain. One is E. coli K and one is E. coli C. Now the bacteria lambda phage, which is grown on E. coli K, we name them lambda K. The bacteriophage which is grown on E. coli C strain named as lambda C. If you believe the previous postulation that phage DNA will be recognized as a foreign DNA, and that will be cleaved by bacterial own restriction endonuclease. Now, if this is the case, the phage, when it enter or infect the bacteria, that will be completely chewed by the bacteria. But then how the phage will survive? What is actually happening? One or two phage will escape somehow, and they will lyse the cell and they will come out from the host. Those phages we named as lambda K as it grown on E. coli K, lambda C in as it is grown on E. coli C. They are, if you plate them, their efficiency of plating is very less, 10 to the power minus 4, because somehow they escape. Now, if you take these phages and then reinfect the same host, you see the cycle, take that lambda K and then again infect the same host, you will see the efficiency of plating EOP is 1. That means first time when the bacteriophage is infecting a bacteria, it will be recognized as a foreign. But if it passes through the host and then the phage which is coming out will not be recognized as a foreign when I am reinfecting them. That time it has the capability to do the lysis of the host. Now, with this observation, what are the interpretation? The interpretations are, it is a host controlled restriction and modification system. The change, that means the modification what is happening in the DNA, because one time it passed through the bacterial host, it now become modified by the bacterial own methylation system. So the change is non-heritable, meaning first time it is modified. Now, if you infect this lambda K to E. coli C, again it will be recognized as a foreign. But once it passed through E. coli C, it will be recognized again lambda C. It will no more be foreign to them. So the changes is non-heritable. Carefully listen, changes is not non-heritable. And the third observation, the phage that survive one cycle of growth on restricted host now can reinfect that host efficiently. Now, the biological role of these restriction enzyme, most bacteria use restriction enzyme as a defense against bacteriophage. The name restriction is actually meant by it restrict the growth of a foreign DNA. Defense against bacteriophage, restriction enzyme prevent the replication of the phage by cleaving its DNA at specific site. The host DNA is protected by methylase, which add methyl group to adenine or cytosine bases within the recognition site, thereby modifying the site and protecting the DNA. Now let us discuss about the sequence where it actually recognizes. Generally, the palindrome is the sequence, palindromic sequence it recognizes. We are discussing about a particular type of restriction enzyme. I will come to the type of restriction enzyme. The mirror-like palindrome in which the same forward and backwards are on a single strand of DNA strand, like GTA, then ATG. If you read GTA in the opposite direction, that is also GTA. Now, the inverted repeat palindrome is also a sequence that reads the same forward and backward, but the forward and backward sequence are found in complementary DNA strand. Actually, a true palindrome, there is a axis which has 2-degree rotational symmetry. Inverted repeat palindromes are more common and have greater biological importance than mirror-like palindrome. See the sequence GAATTC. If you go to the complementary sequence, from the other side, it is also GAATTC. If you can imagine, after GAA a vertical axis, if you rotate two times 90 degree, then it will be the same sequence. That's called palindromic sequence. Most of the restriction enzyme ends of the after cutting produce two types of end. These two cartoons are showing, one is a blunt end, one is producing a sticky end or cohesive end or the overhang. Okay, now the few types we need to discuss here. One is restriction, that is called isocyzomar, neocyzomar and isocadomar. I will just discuss about what is isocyzomar. Restriction enzymes that have the same recognition sequence as well as the same cleavage site are called isocyzomar. And restriction enzyme that have the same recognition site but they cut at the different site within that sequence, we call them neocyzomar. And there is isocadomar, that is restriction enzyme recognizes two different sequence but after cutting they are producing same overhang. Now come to the different type of restriction enzyme. There are four types of restriction enzyme. This is a very examiner's favorite questions. There are four types of restriction enzyme: type 1, type 2, type 3 and type 2s. You need to discuss little bit more not like an undergraduate students what we discuss. Type 1, this is a one enzyme, single enzyme with three different subunit. What are the subunit? First subunit is recognition subunit, second subunit is cleavage subunit, third is methylation specific. They are designated as HSD capital S, HSDR, HSDM. The thing is that they recognize a sequence but they cleave the DNA up to 1000 base pair away. So methylase a single sequence but cleaves a DNA up to 1000 base pair away. Another thing, they modified before it cuts. They recognize asymmetric site. This is not written, I'm just mentioning. This type 1 enzyme, it modified before it cut. Its recognition sequence is an asymmetric sequence. What sequence? Palindromic sequence, which I have shown, that is not the sequence. It recognizes an asymmetric sequence. This type of enzyme required cofactors. It requires ATP and S-adenosyl methionine, SAM, as a methyl donor. Then come type 2 enzyme. You need to remember here it is two different enzyme with both recognition and with both recognition the same target sequence which is symmetrical, that means palindromic. The two enzymes either cleave or modify the recognition sequence. That means two different enzymes recognize same sequence. This is symmetrical. Two enzymes cleaves or modify same sequence. It doesn't require any cofactor like ATP and SAM, S-adenosyl methionine. Type 3 enzyme pretty similar with the type 1 enzyme. It also is one enzyme with two different subunit, recognition modification together and cleavage another one. So one enzyme with two different subunit, one for recognition and modification and one for cleavage. Recognition and methylase same sequence, but the problem is it recognized some sequence but it cut 24 to 26 base pair away. It also recognizes an asymmetric sequence. It also requires cofactor ATP and SAM. Now type 2S, this is also pretty similar with type 2. It is two different enzyme but recognition sequence is asymmetric, not symmetric, not palindromic. So only two is having a symmetric sequence. Cleavage occurs on one side of recognition sequence up to 20 base pair away. So out of these four, we have found that only type 2 are very much useful for recombinant DNA modification manipulation purpose. Now there are many types of restriction enzymes available. One such is very common, eco R1. Okay, fine. Let me tell you, the recognition sequence can be 4 base pair. We tell them tetracutter. It can be 6 base pair recognition sequence, hexacutter. Eco R1 is a hexacutter, that means recognize 6 nucleotide sequence. There are octacutter, which is little rare restriction enzyme. Not one is one example of octacutter. Now how we name them? Abbreviation, meaning E stand for the genus, CO it's from coli, the species, R stand from the strain, which strain they have taken, and Roman 1 it identified the order of identification. This is the sequence I just mentioned. It is a hexacutter, so you can see it recognizes it and produces a sticky end, or what we call that cohesive end. So after cutting, overhang is there, AATT, again AATT. So this is the way it cuts the its target. Now something about DNA methyl transferase. E. coli, there are 3 types of methylase present. One is DAM gene, which actually the DAM methyl transferase transfer the methyl group from S-adenosyl methionine to N6 position of GATC, the A, adenine N6 position. This is a methyl transferase. Another very popular one is DCM. DCM methylase, it is modifies the internal cytosine residue in the sequence CCAGG or CCTGG, the second C N5 positions, not N5, C5 position, C5 position. Now the other one, which is very rare, named as Eco K1, but the site for these enzymes are very much rarer and occur about once every 8 kb, so it's very rare. These are the E. coli methyl transferase we know. There are other methyl transferases also there, which are mammalian. CpG island methyl transferase we will study in eukaryotic gene regulation, how this methylation can regulate eukaryotic gene transcription. This side, left hand side, you need to know this a very important feature in recombinant DNA technology. It is important to eliminate the restriction system and modification system both in E. coli strain to use as a host for recombinant DNA. Now this is a 14 kb, so far I remember 14 kb sequence where the immigration control region of E. coli strain, that means you can see HSDS, HSDM, HSDR, recognition, modification, cut. R stand for cutting, A stand for specific recognition, and M stand for modification. And these are all other related genes which are required for restriction and modification system. In a host E. coli cell where we are going to introduce our manipulated gene should be always R minus, M minus. R stand for restriction site, M stand for methylation. So a wild type E. coli is R plus, M plus. It has both restriction modification system, and the one which are going to use as a host for manipulating our recombinant DNA should be R minus, M minus. Now what about R plus, M minus, or R minus, M plus? Please study and get back to me. Now the application. Isolated restriction enzymes are used to manipulate DNA for different scientific application. They are used to assist insertion of genes into plasmid during gene cloning and protein expression experiment. Restriction enzymes can also be used to distinguish gene allele by specifically recognizing single base change in DNA known as SNPs. We can do gene fragment, gene mapping, actually RFLP studies. There are so many studies we can do by using restriction modification system, restriction enzyme. Now the 2 more other topic I will teach today, that is linker and adapter. see these two things. This is also an examiner favorite question. What is a linker? Okay, linker adapter are short double-stranded DNA molecule, most likely 10 to 12 base pair, that permit different cleavage site to be interconnected. That means the synthetically prepared some of the decameric or this fragment 10 to 12 base pair sequence where some restriction sites we have introduced, and that need to be ligated to your gene of interest or during manipulation. See this diagram carefully. This is a decameric linker molecule. That means synthetically oligonucleotides we have introduced. What is there? An EcoR1 site you can see there. Now this purple color one is our gene interest. That means insert. That means foreign DNA, which I want to clone in a vector which I already cut with EcoR1. But unfortunately, my insert is a blunt ended one. That means it doesn't have any sticky end. But I want to clone where? In a vector which is already cut with EcoR1. In this situation, how I am going to ligate my foreign DNA to this vector? One such way is, now use this linker molecule which are already having EcoR1 site designed, already pre-designed. Now I can use T4 DNA ligase which will help blunted ligation, and I can ligate my gene of interest, that means foreign DNA, with the linker molecule. Why two products? Products could be, they are linker, they can join one by one after another also. Now how do you know which one is your desired size? Because you know this is my foreign insert size. If I add linker, it will be the correct size. So you need to run a gel and need to fractionize that this is the size. Then now you can cut with EcoR1 because your linker is having the EcoR1 site. This thing you can now ligate in the vector. Now the question is that if you want to take your only the foreign gene of interest, how you are going to do? Generally, the linker and the vector where it is actually joining, that site, it will generate another restriction site. With the help of that, you can take your foreign DNA. Try to understand the question. If I cut with EcoR1, I can only get back my gene of interest with the linker, but I want only my foreign DNA, that is only the purple color one. How do I get it? Answer is, once it is joined, at the joining site, you need to design such a way that another restriction site will be developed. Our next discussion is adapter. Now adapter is also a synthetically designed double-stranded DNA molecule. Why we need to use adapter? What would happen if the restriction enzyme used to generate the cohesive end in the linker will also cut the foreign DNA at the internal site. Now you imagine a situation, the enzyme you wanted to clone, that site is already there inside the internal site of your foreign DNA. Then if you use that restriction enzyme, your foreign gene will be two pieces. So you don't want that kind of enzyme. You can change the enzyme. If you don't get, then how you are going to clone? See the problem, okay? Now this is also an adapter molecule. Carefully see the 5 prime of this one. It is already having a BAMH1 sticky overhang already synthesized, so that I can get, this is after BAMH1 digestion, I am getting this kind of sticky end, and the 5 prime site is already hydroxylated. Now this molecule I want to attach with my gene of interest with the help of T4 DNA ligase and ATP. Now this is my the product. After that, all are hydroxylated. So this, why hydroxylated? So that self reduce the chance of self ligation. Now what will happen? Polynucleotide kinase, PNK. I'm giving another enzyme. You remember I already taught you the role of PNK. It can end labeling. It label the 5' phosphate group. Now with this, now you can use the vector which is already treated with BAMH1. Okay, and then can be linked with your vector. Let me read what I explain here. Use of BAMH1 adapter molecule. So this is a BAMH1 digested, already by BAMH1 digested, synthetically developed oligomer. A synthetic adapter molecule is ligated to the foreign DNA. The adapter is used in the 5' hydroxyl form to prevent self-polymerization. The foreign DNA plus ligated adapter is phosphorylated at 5' terminal and ligated into vector previously cut with BAMH1. That means your vector is also treated with BAMH1. You can get your clone done. Now this adapter and linker are very, very important. These are also examiner's favorite choice. So please go through, practice it. Any problem, any question, please get back to me, call me. I will explain it. Thank you very much and for today.